Um, okay, hello everybody, and it's my pleasure to introduce Professor Sayan Mukherjee. I wish both of us were there right now, but unfortunately, circumstances dictate otherwise. I have known him from, I think, 2004 or 2005 when he joined as a faculty member at Duke. I was a graduate student, and I very fondly remembered sitting through his lectures on a statistics course. And so basically his research is in statistics and machine learning. And he applies them to a really wide range of problems over time, right? He's gone from biology to geometry now. And I think he's still at Duke, but right. Just last year, I believe he won this Alexander von Humboldt professorship in AI, which is a very coveted research award in Germany. And since then, he has been at Max Planck Institute for mathematics in the sciences, and he's at Leipzig, right? Right now, Sayan? Yes. Okay.
I am physically right now in Leipzig.
Okay, good. And so he's going to tell us, talk to us about modeling count data, noisy count data. Yeah.
Okay, so today is count data 1, and I think I'm giving you a count data 2, and I think the third talk is on imaging, 3D imaging of shapes. So a lot of this is joint work with Justin Silverman, who is at Penn State, Lawrence David, who's at Duke, and Kim Roche, who is just recently graduated. So let me start. Let's see. Okay. So I'm going to start with the example of microbial data. So what is a microbiota? It's a collection of microbes associated with a host. There are about 10 trillion bacteria in our bodies. What do people consider the microbiome? The collection of microbial genes associated with a host. This slide just says that people have been thinking about the microbiome for a while. It's just the scale has been different, right? And now we can measure so many of these bacteria. And you can look at it in different parts of the body. Okay, so what I'm going to talk about today is methodology, right? And what I'm really going to talk about today is how do we think about understanding variation in really noisy count data, and what are the challenges in understanding variation in really noisy count data. Okay, and an example we're going to look at is what's called a bioreactor, right? Which is a fancy glass bottle where you can measure a lot of things, and in this case you can put in poop, and you can put in microbial specimens and see how things change. Okay, so something that I think is really important when we think about modeling, right, is what is the model for data collection and what is a model for sample processing, right? So you collect the data, you sequence the data, you assign sequences to samples, you denoise it, and you make a count table. Now, the thing is, there's so much noise in all of this, right? So in sequencing, there's problems with counting and bias in terms of random subsampling. There's the issue of technical variation and bias, again, random sampling. And there's a question of how much of what you're seeing is biological variation versus just signal. And so this is an example of the data you get, but this is a real data. It's amazingly sparse. And in the second lecture I'm going to give on noisy count data, we will talk about how the sparsity can really affect conclusions. Okay. So there's been kind of two camps in really noisy count data. And one is that the data is compositional, and the other one is it's not. These are counts. And so let's go through that in some detail, okay? So what I'm showing you now is and if anyone has questions, please ask me. What I'm showing you now is a simplex. What's a simplex? Things add up to a constant, okay? And that is the compositional view. And what I just did was write out the math equation for a simplex. Okay. Now, there's a beautiful connection between the multivariate normal distribution and geometry. So if you have something that's multivariate normal, you have statistical independence, means orthogonal bases. And you can actually write out the variant, the mean and the covariance just using straight up linear algebra, which is really nice. Okay. I'm just showing you a picture of PCA. PCA is a classical example of how the multivariate normal gives you something that is really intuitive, and you can relate the statistics to the geometry of the data. Okay. Now, what else is nice with the multivariate normal or with normal data is that if you know how your treatment effect is pushing something, right? It's a linear map. And that is so useful and nice. Now, when you are on a simplex, all of that falls apart. And the reason why it all falls apart is that everything has to add up to a constant. So if you have these three different bugs, right? They all have to add up to a constant because it's compositional data. And the problem this causes is that if one goes down, or if you introduce a treatment that makes one go down, the other ones are going to go up. So the coordinates are no longer independent. And that makes things a ton more complicated. Right? So if we go back to the previous case, you don't have a linear transform. And what this induces is a negative bias. And this negative bias, if things are not sparse, aren't terrible. If things are sparse, it's really, really bad, right? And again, this is the idea that comes from compositional data. So in compositional data, the axioms are scale invariance, permutation invariance. You can reorder the bacteria, it shouldn't matter, and subcompositional coherence. which is if you take a subset of the data, your analysis should not contradict what you have. Okay, so in compositional data analysis, there's something called the CLR transform. The idea is you take you take SD and you map it to RD, and you're just basically taking logs of all of the components with a geometric mean to normalize. Okay, now the compositions are normal if the CLR from one coordinate to another coordinate are independent. And there's something called the basis contrast matrix. And I'll come back to this in a second. There's another form of transform, which is called the ILR transform, where you take the CLR transform and you weight it by a matrix. Okay, so if you look at the inverse map of the ILR transform, it rewrites as this exponentiation. Now, one thing that we did that's interesting is that you can, if you know the phylogeny of the bacterial species, right? Then you can actually write out what phi is. You can write out the basis contrast matrix. And I'll stop on this for a second. But it's really a question of the nodes, right? Of the taxa. Now, what's interesting is with the ILR transform on the left, this is what we would have in the simplex. And with this new coordinate representation, we come back to the Gaussian world. Right? And so the point that I'm trying to make, and a point that I've done in a lot of my work, is that are there representations of the data and transformations that make them very amenable to either statistical algorithms or deep learning algorithms, right? And again, this is an example of that. Okay. What else is interesting is if you look at what's going on in the simplex in terms of, let's say, one species is growing, the other one is decaying, right? And these are exponential, right? Once you transform them, they're straight lines. So linear models work really well. Okay. So the broader idea is that you have unobserved absolute abundances, right? This lives on a simplex. You transform this through the phylogenetic tree, and then you come up with a new space. Okay. But there's a really basic problem with the compositional perspective, which is these data are sparse. So when you have a zero, you get negative infinity. And well, that's a problem. And it's also true, the data do really come as counts. They might be super noisy counts, but they do come as counts. So again, let's go back to the basic picture. You collect data. And what are you doing? You're just grabbing from a bag, right? And let's say we have system 1 and system 2, and they both have very similar distributions of what's in the bag, right? But because of their size, right, something very different could happen, right? So random sampling induces competition to be counted. This is called count compositional. So the question is, can we pull out more information from counts? There's a really cool, oh, shoot, I wish I had it here. Picture from a book by Andy Gelman on stomach cancer. So actually, let me just step back a bit. Does anyone know what I'm talking about? Please, someone say something.
So I think it is pitched a little bit above what we've been seeing so far. So I don't, I'm also getting slightly lost with the.
Okay, so what the data was a distribution of cancer across counties in the United States. Okay, and the first picture was that counties that were very sparse had a higher percentage ratio of cancer. Okay.
Can you roll back to the slide where you had the two samples, that checkerboard kind of slide where?
Which one?
I think
This
it's
one.
2 slides ago, that one.
This one or one before?
Oh, this one.
Yeah.
So
Okay, so what I'm trying to tell you, go ahead.
You know, you said, I think system 1 and system 2 had similar compositions of the 3 species, but you're saying because it's a small random sample, I don't think I understood the line.
Yeah, that's exactly right. This is exactly right. System 1 is a small random sample, right? System 2 is again a small random sample, but of a bigger collection, right? And so the point I'm trying to make is when you sample things, right, and there is a competition between the objects, these are no longer IID, right? There is a compositional aspect of it. But there's also an extra problem, right, which is we don't know the total counts.
So there's one more question here.
Mm-hmm. Okay.
Hi, Sayan. You said system 1 and system 2 has similar distributions, and you just pick same size of samples from both system 1 and system 2. What do you mean by similar distribution? Like, how do you?
So let's say they were the same identical distribution. Okay. The question is, I'm sorry?
So if they are identical or similar distributions, then what we want to conclude from the small samples we are picking, if we already know the distributions are
so
similar.
yeah. So the point I'm trying to make is variance matters. Right. And if you're looking at noisy count data, right, and you have a smaller sample, it's going to have more variance. And that can change your conclusions a lot. right? And the other part of this point is that normally in a statistical analysis, we have control over how big the sample size is and you know, how many total coin counts we have. But here, in a lot of these type of noisy count data, we don't have that control, right? Because of how the data is being generated and all of the aspects of the noisiness. And so that's what I'm trying to get through in this slide.
Any other questions before we move on?
Yes, one more. About the simplex thing. So you start with simplex and then you transform it to another coordinate system. So can you please tell more about, like, why do you start with simplex and then go to then do one more transformation? More details, maybe little.
Yeah. I'll actually go talk about that in a lot more detail soon. But the idea is on this slide, right? Is what I'm showing you here is hard to model. And I'll tell you very soon why it's hard to model, right? But once you transform it, it just becomes Gaussian. And so the argument is if you think, and we'll talk about this in more detail. If you really believe your data is compositional, right? Then what you can do is you can use these transforms and you get to something which is just multivariate normal. So it's a mixture of Gaussians, right? And I'm sorry?
What is this transformation, like going from simplex to another coordinate system? What transformation you do?
It's this transformation. Well, it's a step. Yeah. So this is a transformation. So what you're doing is you are first taking all of your counts, right? Normalizing it by the geometric mean and taking the log of it. Okay?
Yeah.
Now, the problem with that is that it does not give you a coordinate system that is orthonormal, right? Which is what's really nice about Gaussian. Now, you can make it orthonormal if you rotate it by these fees. But the question is, what are these fees? And what we can show you is that if you give us the taxa, if you give us the phylogenetics, we can give you an algorithm to generate the fees, right? And now you have an orthonormal system. And that's what lets us take these isoclines, right, on the simplex into a Gaussian.
So I have
Okay.
a very basic question, that is it a simplex because of resource shortage? So I mean, one species can only grow at the expense of the other.
That's exactly right.
And the sparseness is the sampling, not the population itself.
That's exactly right.
Right.
Yep.
Okay. Thank you.
Yep. Exactly. Thank you. And again, this is showing you exponential growth and decay in that space. Okay. So what now I'm trying to tell you, right, is that there's something that lives in between. You can't just think of this as purely compositional, right? But the counts are super noisy. And the question is, why are the counts super noisy? The counts are super noisy because you're doing random subsampling, right? And that's very basic in the process. the less sparse your data is, the less this is of an issue. The more sparse your data is, the more you have a problem. Okay. Give me 1 second. Okay. So we'll go through this fast because I think most of you know this. So basic Bayesian statistics, right? You have a likelihood. And what you are looking at are the probability of the data, given data, that's your likelihood. And then you have a prior. And then you use that and conditional probabilities to get a posterior. And that's what I just wrote down. So what I'm writing down now is something that, for lack of better words, interpolates between the compositional perspective and the count perspective. It handles zeros. It allows positive and negative covariation, and it models multiplicative errors, okay? So the classic way people have tried to model count data is through something called the logistic normal, which I'll show you in 2 minutes. So your data Y is multinomial, given the probability distributions, and pi, the probability distribution, is what's called logistic normal. Now, what I'm going to advocate for is, instead of doing it this way, do the same thing with Y, but do a transformation on the pi. And that transformation will let us go back to the multivariate normal. And there are some good arguments for why you want to do that. Okay. So this is the logistic normal distribution. And the problem with the logistic normal distribution is a normalization. It's the product over I, Y, I. That makes it very hard to sample from. That makes it very hard to compute. That also makes it very hard to even do maximum likelihood estimation. Okay. So what we worked through and observed is that this thing here, can you guys see my mouse?
Yes, we can see it.
Yeah, okay. This thing here on the left is a classical way people have modeled noisy count data, right? You have a multinomial and the probabilities are logistic normal. And what we're advocating is what's on the right. And the reason why we're advocating what's on the right is that Ada is much easier to model than pi. Because it's just straight up multivariate normal. And sampling is easier if you want to do MCMC. Even MLE is easier. Okay. So let me talk you through an example, okay? Okay. This goes back to the bioreactor. You have that glass object, and you have put microbial sample in there, and maybe what you're going to do is hit it with a probiotic, right? And you want to know how much it changes and how it's changing. Okay. So in terms of the model, you have the true state, which we don't know, right? And this is how you are seeing the biological variation. And then you're injecting these W's, right? Which are your biological variation. These are your probiotics that you're injecting. Then you have technical noise, right? Because you always have technical noise. And then you have your observed counts, which are super noisy, right? And in a way, what you're really interested in is breaking it down the W and the V. You want to break down the biological variation versus a technical variation. Okay? So this is your model. And this is an adaptation of something called a dynamic linear model, which is used in finance a lot, right? For count data. You can also think of this as a discrete Kalman filter, right? So what's happening? Your observed counts are multinomial based on these probabilities. These probabilities pi are given from your from the inverse transform, right, of these etas. These etas are being updated linearly, right? In terms of the technical noise, and then the underlying states theta are being again updated linearly, right? The only thing that is different from this model in a common filter, well, there are a few things. The main thing is that it's not homogeneous as time changes. These matrices change, number one. And number two, we're using this deterministic transform to make the problem easier. So we call this a multinomial logistic normal dynamic linear model. And so, okay, let's talk this through. So we have a longitudinal model with repeated observations. Here's the model. These are our daily samples. And there's something that's really important here, which is one of the things we really wanted to understand when we were working on this project is at what level of time resolution do we have more technical variation versus biological variation, right? Another version of this is, how often do you want to sample the patients, right? And the question is, how do I set up an experimental design to be able to do that? And if you want to do that, you can't just set up a straight up time series design. What you need to do is you need to have multiple multiple glasses, I guess is the right word. And every once in a while sample in parallel, right? So it's this idea that you're not running just one time series, you're running a few time series, and every once in a while we do replicate sampling of the time series. So we can get a better idea of what the technical variance is. And with this, with these longitudinal samples and the replicate samples, so the longitudinal samples give us a notion of what the biological variation is, plus the technical variation. The replicate samples give us an estimate of the technical variation. And so this is what we set up. We had 20 replicate samples. And so just to show you some simulated data, these are simulated observed counts. Our phylogeny. And this is our posterior estimate of the theta, which are the probabilities, right? And the other thing that's nice is we can give you uncertainty estimates on these. So what else can we do? We can decompose this. We can decompose what we are doing into the biological variation, W, and the technical variation, V. And again, as a picture, we can show you what is going on in terms of the biological variation, the technical variation, and the total variation.
Question, if you could go back a couple of slides.
Yeah, sure.
Yeah. So these are simulated observed counts and posterior estimates for theta. Okay, I missed the true value. So
yeah.
All right.
I'll show you real data as well soon.
Yes, yes. No, I was interested in how well you're doing with the synthetic data. Is it that I can't see a blue line? The blue line is exactly on top of the green line?
That's exactly right.
Okay. Okay, thanks. Yeah. One.
Yeah. Is that better?
Yes. Yeah, go ahead. There's a question back there.
So it was also interesting. One of the things we found is that when some of the vessels.
There's another question at the back. Sorry.
Oh, sorry. Go ahead. Let me look in the.
Sir, so what is is on the y-axis? So can you show that graph, that posterior one?
Well, this one? No. Sorry.
This one. Not this one. I think, yeah, this one. What is this balance value?
Yeah, okay. So actually give me 1 second so I can get my charger. The balance value is the renormalized value of the estimated species counts.
Okay, so can we understand it is a standard deviation or in terms of standard deviation? So how much, how much standard deviation it's away from the mean value? Is it?
What it is, okay, let me restate this. Okay. When you're doing Kalman filtering, right, or you're doing some hidden Markov model, what are you doing? You're basically doing some smoothing, right?
Mm-hmm.
When you're doing filtering, you're doing smoothing. So the orange values are kind of your raw data.
Okay.
Okay. And then let me move forward one. Okay. Okay. So what we have is actually the green values are your true numbers. Okay.
Okay. So you normalize these values, these count values.
We smooth them using this algorithm. Yeah.
Okay, so it's another way of transforming count data.
It's another way of transforming count data. And the idea is this filtering or this smoothing gives us the, what are they, the true underlying state.
Okay, to combine both kind of variances, right?
That's exactly right. That's exactly right.
Okay, thank you.
No problem. And then the question is, can you break apart the two types of variances? Right? And that's what we get to here. So what we can give you is a matrix of how the different proportions are covariant. Okay, and that's what this picture is trying to show. And what's interesting is you can use this to see, for example, here something happened at this point, and there was a feed disruption of the probiotic. So you can start seeing things that are changing. And we've used these ideas as well to ask about how in baboons, are there universal rules of microbiota that are changing, or is it specific to individual baboons, or is it specific to troops? So you can use, again, these tools for asking questions like that.
Sean, maybe you can go back to presentation mode.
Oh, sure. Sorry about that. Yeah. Give me 1 second. We're back?
No, yeah. No,
Okay.
in presentation mode. Yeah, this is fine. Thanks.
Okay. So again, we can give you these estimates of confident or credible intervals. So then you can start asking questions. Are there sub-daily dynamics? How fine is it? Right? And the point here is because we every once in a while collected data across a bunch of these for lack of a better term, glass objects, right? We can actually give you an idea of what the subdaily dynamics is. And we can tell you at what point does a biological variation exceed the technical variation. And I think that's actually a useful experimental design, right? To try to get at the question of how fine you need to sample. Okay, so when we first did this, we did not have a fast algorithm. And the reason we did not have a fast algorithm is that the sampling algorithm to do the Markov chain Monte Carlo was super slow. And it's because the marginal distribution was complicated. So what we worked on after that is a class of models that are way, way, way easier to sample from. And so before I go into this, let me give you a highlight of the next count lecture I'm going to give, which is going to talk about under what conditions and what assumptions can we get differential expression from very noisy count data? Okay, but let's start with this again. So we introduced something called the marginally latent matrix T processes. And these are Bayesian hierarchical models. And so on the left, we have a conjugate linear model. On the right, we have a dynamic linear model. Okay. These models have the same marginal form. And what we can show is that we can come up with a really good approximation for the multinomial. And that's basically what we did. So we basically came up with something that was 5 times faster than HMC and faster than variational Bayes, but more importantly, was way more accurate than variational Bayes. And unless your data is crazy sparse, we do well. And this is the paper. And basically what it ended up being was a uncollapsed sampler. Let me go through this quickly because yeah. I can give you more details. The paper is now out in JMLR. But let's talk rather than me talking more.
Maybe if anyone else has any questions or clarifications, this is a good time. And also there are delays as we pass around the mic in this room so that Sayan can hear the questioner. So excuse that. Anybody else? Yeah, go ahead.
So you are collecting data from patients, or you're just doing experiments on microbial communities with the probiotics I did?
Right. So this is a very good question. So what I talked to you about and I showed you in these slides are from these bioreactors. And we did it in the bioreactor because everything was extremely controlled. And we knew what was going on and we knew what we did. Now, we have also done this in patients. We have done this in children's microbiomes. We've done this in vaginal microbiomes. And I think they're not patients, but we've also done this in baboons.
In human being, I guess the environment is not constant, for a microbial.
I'm sorry?
Like if you are selecting human stomach, if you are experimenting probiotics on human, so in that case, the environment for the microbial community is not constant throughout. It may change by different food and if you're collecting data on time basis.
That is a great question. So both in humans and baboons, we've had to detrend in terms of, it was easier in the baboons because we knew when the dry season was and when the wet season was, right? But in both cases, you do have to detrend.
Interactions between the microbial species. I mean, over time, if the microbial community is changing, the interactions are also changing. Okay, so can we extract information about the interactions, like overall, these many species are there and they have such interactions between pairwise interactions I'm talking about.
Yeah, totally.
Order interactions.
Yeah, so higher order interactions, I don't know. We haven't been able to do that yet. pairwise interactions, that's something we actually did. And we studied, in humans, it's been harder. And the baboons, it was a little bit easier. And we actually have a paper on bioRxiv, and we have studied how similar are the microbial interactions across different animals, right? And once you detrend, what you find is that they're very similar. Now, there's a difference in that humans, right, get different health. The variation of the of what you can do in terms of health in humans is a lot different than in a baboon, right? So we used the baboon system because we knew things were much more uniform in terms of the environment. And what I don't know, and I would really like to understand, is how variation in the environment affects these interactions. And that I don't know how to answer.
You must know. In introducing probiotics in an existing community, it may change the overall interactions, or it may eliminate some of the species as well. So actually, I was talking about pairwise interactions and how the species are also evolving. I mean, the pairwise interactions between, let's suppose there are two species, A and B, in a community at time t, and when we introduce probiotics at time t plus some delta t, so does this pairwise interaction also changes over time, or not? It's constant over time. Like I'm talking about higher order interactions.
I totally understand what you're saying now. Yes, it does. It does in a bioreactor, that I know. How it changes in a human population, in like a real data set, I don't know, because I haven't been able to set up the control in terms of in terms of technical variation.
Okay, thank you. Anyone else? So just in the interests of pedagogy, I was wondering, can you explain what is the logistic normal distribution? And I realize without a whiteboard, maybe it's difficult, but I mean, people here are familiar with the normal and with the multivariate normal and so on, but some of these
let's go back. Yeah. Let's do it.
Right.
Give me 1 second and we'll do it. So the logistic normal distribution, if you look at it, this entire part is just log things and it's normal. So it's not a problem. Right? Does that make sense to people?
Why is it a normal distribution? I mean, the log is there, but there's no.
The log is there, but once you do this transform, right, it's, you know.
Oh, okay, sorry. I missed that it's a matrix. Sorry. Yeah.
Yeah. Okay.
Okay. Yeah.
The problem with the logistic normal distribution is well, give me a second, is that part, the product over IYI.
Okay, and
the question is, why is that a problem?
Actually, I didn't understand what the argument of the log, that y from 1 to d minus 1 divided by y of d. Where does that come from?
So what you're doing is you have a simplex, right? You normalize, take the log,
yeah. Right.
make it a normal.
Okay.
Okay. And that's what this part is doing. Okay. And so
Okay.
that's fine. That's not the problem. The problem is the normalization constant. Like
So-
in much of life in MCMC, the normalization constant is a problem
Okay.
because you have to normalize by the total counts or the geometric product of the total counts. And that's what makes this hard to sample from.
Right. So I probably have to sort of read your reprint and so on to and papers too.
But
There's
yeah,
one more question back there.
sure.
Sir, so here, what I understood, are you assuming that the distribution of Y as a normal, but in the count data, it can't be normal. So that's the problem in this thing.
That's the whole problem is this thing, right? So
Yeah, because we don't know the distribution of this Y, and here we need to assume the distribution of Y as a normal. And that's
So-
why this distribution is means this method is not appropriate for dealing with this kind of data.
So yeah, so if you just take noisy count data and apply standard statistical methods, the problem is a lot of them are meant for normal data, right? So you have a problem. Now you can use this logistic normal model, right? In theory, but the problem with this logistic normal model is inference methods with this logistic normal model are, number one, very brittle, and number two, super slow. And a lot of that is because in the denominator, you see this product over IYI, right? And that normalization constant slows everything down a lot. And so that's a problem. I mean, so another way of saying this, right, is if your data weren't very sparse and look kind of Gaussian, right? Everything I told you, ignore. Just run a normal model. Right? Just do whatever you want, use a deep neural network, everything will be fine. Well, kind of. But if your data are sparse, and the sparser it gets, the bigger of a problem you have if you try to do that.
Okay, so I think we can continue tomorrow. Thank you very much, Sayan, for this talk, which was perhaps a bit challenging for some of us, but we should go back and read your material and work.
I sent the slides, I hope.
Sorry?
I sent the slide, I hope.
You might have, I'll check.
Okay, if not, tell me, I'll send it again, okay?
Right, yeah, if you send it to
And
program,
then-
they'll put it on the website.
Okay, and then tomorrow, we'll do it again.
Yes. Right.
Thank you.
And so thank you, and thank you, Leelavati, also for the introduction. And right.